Today, we're going to be talking about meiosis, the other version of mitosis that produces 4 cells. Meiosis is the process of gametes, which are basically sperm and egg cells, dividing to create more sperm and egg cells, specifically 4 of them, because each of them will have a quarter of the original's DNA. So basically, it's like mixed up sorta. And I'm going to make a white screen appear. Boom. So you can see the illustrations better. Okay, let's start with one cell. This cell is the mother cell, and the cell that it will divide to will become the daughter cells, and then they will divide too. Meiosis is very similar to mitosis. So let's start off with phase 1 of meiosis. Prophase 1. You realize there's little 1 at the end. Well, that's because later it's going to happen again. Prophase 1 is actually divided into 3 phases: early prophase 1, middle prophase 1, and late prophase 1. Meanwhile, mitosis, the one for normal cells, only have 2 prophases, early and late. Early prophase 1. It is basically the same as in mitosis. The chromatin will clone itself and the same pairs of DNA will flow together and form a chromosome. And that's basically literally exactly the same. So because that's boring, let's move on to middle prophase 1. Here's where something happens, something exciting. Synapses will happen, where homologous chromosome pairs will pair up, and each pair is called a bivalent. This is very important for the future stages because it creates diversity later on. And a homologous pair is basically a pair that has around the same genetic material, size, width, and height. So it finds its twin, basically. And then it will do late prophase number 1. This is where crossing over of chromatids can occur. This will create a large amount of diversity. It can swap its hair colors, it can swap body height, body weight. It can also cause deadly diseases to occur, which is not very good. It can change hair color, it can change nose shape, ear width, thickness. It can change everything. Basically, this determines the characteristics of the baby. And then, let's move on to the second stage. Metaphase 1. The chromosomes will line up in pairs, so there's 2 lines of chromosomes instead of the usual 1 in mitosis. And these pairs of chromosomes will attach themselves to the spindle while also being in the middle. Well, that's basically the entirety of metaphase 1. So let's move on to anaphase 1, where the chromosomes are pulled to opposite sides of the cell by the spindle. The spindle will retract and they will get split together. So they will be pulled apart. It's not really breaking the chromosomes, it's just pulling them apart from their homologous pair. And then the cell splits as usual in telophase number 1. And as for the 2s, which is prophase 2 to telophase 2, it will perform normal, regular, same old mitosis. It won't be exactly the same as in meiosis because it doesn't do middle prophase and it also doesn't do crossing over. So it's basically mitosis for both of the daughter cells. And finally, we reach the last phase, the limbo interphase. In this phase, the gametes will spend most of their lives, and they have a chance to fertilize or be fertilized by the other gamete. So a sperm will fertilize the egg, and the egg will be fertilized by the sperm. And depending on which sperm will be the lucky one to fertilize the egg, it will result in the baby's characteristics. And that's basically all of meiosis explained in one video. Thanks for watching. Bye!